सर आप हर बार हमारी यूनिवर्सिटी का नाम बदल देते हैं।
किरण एंड पल्लवी पटेल है ना?
किरण एंड पल्लवी पटेल ग्लोबल यूनिवर्सिटी केपीजीयू।
केपीजीयू तो बोला मैंने मैडम।
वो आपके केजीपीयू बोल रहे थे।
अच्छा।
तो वो थोड़ा हो जाता है, चलेगा।
ग्लोबल में पहले ले आता हूँ। पल्लवी मैडम
आप हमें ग्लोबल पहले बनाना चाहते हो, इसीलिए ऐसा हो जाता है।
Yes.
Fine, sir. Okay, so welcome all the participants and welcome to the speaker, Varsha Madam. So it is my pleasure to introduce everyone to the today's speaker. But before that, just a brief introduction about this series. This is a talk on RT-PCR based diagnostic for the infectious disease, which is under the banner of this Science Dialogue series, which is being initiated by Gopal ji sir a few months back. And since then, many individual speaker, rather saying many experts, they have delivered a lot of very good talks, which were really benefiting for the students. They were from the academia, but now probably, sir, this is the second talk which is from the industry or some kind of other than the academy, because one was from the industry at Gandhinagar probably.
हाँ इन ग्रेस। वैसे इंडस्ट्री वाले तो बहुत सारे लोगों ने किया है, बट यस प्योर इंडस्ट्री के जो
प्योर इंडस्ट्री this is the second one which is now we are arranging, which you have arranged for the students. So this is the second one. So I welcome Dr. Varsha for this Science Dialogue series. So for the introduction to the participants, she is a PhD in biochemistry with a specialization in structural biology from the great MS University of the Baroda. And currently she is holding a kind of specialization, or better to say, she is now leading the molecular biology diagnostic department in the Unipath laboratory, where she has established a cutting-edge molecular biology lab. They have secured an NABL accreditation, which itself signifies, or which itself specifies her contribution to the laboratory, as well as what kind of extensive work they have done for the accreditations. So currently she is overseeing a team of 5 to 7 professionals, where she is trying to ensure the rigorous quality standards, so that whatever the results they are getting through the molecular diagnostic, they will be pursued for the sure shot for the patients also. Now for the last 3 years, they have conducted a substantial number of diagnostic tests, molecular diagnostic tests for multiple infectious disease. Thereby, they are trying to impact the overall patient outcome, so that they may get a better suited results for them. So I welcome Dr. Varsha for today's session, which is on this RT-PCR based diagnosis for infectious disease. So I will hand over this virtual platform to you, ma'am, so that you can initiate the talk, and our participant can get benefit from your expertise.
Good evening, everyone. Thank you, Poonam ma'am, for this wonderful introduction. And I'm really grateful to Gopalji sir for giving me this chance to present this topic today. And thank you everyone for joining us today.
फुल स्लाइड मोड में कर लीजिए इसको।
So I'll be talking on RT-PCR based diagnostics for infectious diseases. I hope my slide is visible to everyone.
फुल स्क्रीन मोड में नहीं है।
Talking on RT-PCR based diagnostics for infectious diseases. So I hope everyone is aware of the term infectious diseases. Now, these are the disorders caused by bacteria, microorganisms, or microbes such as bacteria, viruses, protozoa, fungi, or parasites. Now, there are trillions of microbes present, but only several thousands of them are disease-causing, also known as pathogens. In fact, many species live inside human body or on human body.
एक मिनट में मोबाइल से कनेक्ट कर ले रहा ताकि जो आ रहे हैं उनको देख पाऊँ। फुल स्लाइड मोड में मैं देख नहीं पा रहा था ना कौन आ रहे हैं। So now I can allow admit. Yes, madam. Please continue.
So I was saying, in fact, many many of the organisms live on human body or inside the human body, consisting of the normal flora, and thereby protecting the human host from invasion of pathogens. Now, infectious diseases are a leading cause of death worldwide. Sir, first slide. Okay, sorry, second slide. Yeah. Now, infectious diseases are the leading cause of death worldwide. The 21st century has witnessed severe outbreaks. By this, I do not mean to say only about the COVID-19 pandemic, but also SARS, COVID, MERS, COVID, swine flu, Ebola, and Zika viruses. And as we all know, now recently the swine flu cases are increasing, and Gujarat being ranked at the fourth position currently. So, out of top 10 causes of death reported by WHO, the five are due to infectious diseases. The top single killer agents being HIV, malaria, and TB, and the rest are due to acute diseases and diarrheal infections caused by various agents. Now, in India, in 2021, pneumonia emerged to be as the primary cause of death, claiming over 14,000 lives, followed by acute respiratory infection, claiming over 9,000 lives during the same time period. And by 2025, as per PM Modi's vision, we plan to fight against TB and rule it out, eliminate it. So, as India deals with both communicable and non-communicable diseases. So next slide, please.
Changed.
Yeah, thank you. As India deals with both communicable and non-communicable diseases, the government and private healthcare sectors, they plan to give a better health sector environment. And by this, they do not only plan to treat the illness, but they also plan that humans live a longer lifespan in a healthy way. So, for many infectious diseases like COVID-19, dengue, chikungunya, influenza viruses, HIV, early treatment and diagnosis is of prime importance, or you can transmission of it. Also, they are helpful in public health surveillances, resource allocation, and emergency preparedness.
Next. Yeah.
And
Yeah.
so, sir, एक slide teacher. So one side back.
आपकी आवाज नहीं आ रही है?
Previous slide.
हेलो.
Sir, previous slide.
आप लोगों को मैडम की आवाज आ रही है क्या?
Yes, sir.
Yes, sir. Yes, sir.
हेलो. पूनम मैडम.
हेलो.
हाँ सर, आ रही है.
ओके, तो मुझे नहीं आवाज आ रही है, इसका मतलब क्या?
आपने वो थोड़ा वॉल्यूम धीमे किया है इस वजह से.
ओके. ओ भाई, समथिंग. प्लीज कंटिन्यू.
सर, एक स्लाइड पीछे, previous slide please.
हेलो.
Okay.
आवाज आ रही है?
Yeah. Okay.
आपकी आवाज मुझे नहीं आ रही है.
I'll just continue. Although the diagnosis of infectious diseases traditionally relied on culture-based methods or microscopic examination.
ठीक है. Please continue.
But recently, novel techniques which include speed, sensitivity, and specificity like RT-PCR are now being made into use, which I'll be focusing on today. Now, typing infectious diseases requires additionally serological techniques or molecular techniques. Now, these studies are of prime importance for epidemiological surveys, disease progression, and establishing the causality between a disease and microbial agents. To sum up, infectious disease testing is really crucial, but each of the method faces a challenges. Like if we go for culturing system, they can be limited to bacterial targets, and we can miss the pathogen organisms. also the results take too long to show up.
Yes, slide.
There are challenges. Sir.
Okay.
And there are challenges with the molecular test as well, that they have a fixed content, and we cannot include immediately the concern pathogens of concern. The next slide, please. So for these kits, they are become a popular choice in diagnostic field. We look in also the fast around time, and we can perform control, and the workflows are really simple, and it has automation and high throughput capabilities. So RT-PCR test, by the RT-PCR test, we can diagnose and detect viral pathogens such as influenza viruses, hepatitis viruses, and many others, and bacterial infections such as TB. Also, viral load estimations could be done. Also, we can do the subtyping and genotyping of viruses, and we can also detect antimicrobial resistance genes. Many universities currently are enrolled into this project in along with the GBRC. So recent developments in the field of medical testing and especially molecular biology have been quite amazing. Number one, we all have the facility of POCT, that is point of care testing. Now, by this, I mean the testing could be done nearby to the patients, like in a clinical setup as well, in the clinic. So this provides us a timely decision making, and which is really crucial in times of like we have a COVID breakout or pandemic situations, which saves our time. Now, traditionally, the RT-PCR machines were very large, huge, and bulky, and they required consistent connections. So, to tackle these issues, nowadays portable battery-operated RT-PCR machines are available. As you can see over here, this red one is 96 well MIC PCR machine. It is based in an Australian-based company has developed this, and also the use of AI into the analysis part. It really is of great help. The software can perform analysis over large data sets, and thus that it provides consistency and saves a lot of time. Also, the implementation and integration of microfluidics has helped us for sample preparation and amplification of nucleic acids. So, RT-PCR gained prominence during COVID-19 pandemic, and proving to be a valuable asset during the challenging times. And it also had a great impact on public health interventions and surveillance strategies. So, what is RT-PCR? I assume everyone might be aware of the term, but still, let's have a look at the principle. So, the key techniques involved in reverse transcription and the real-time PCR are total RNA isolation, reverse transcription, and PCR. So, reverse transcription synthesizes cDNA from uh, DNA using DNA as a template with the help of reverse transcriptase enzyme. And further PCR cycles can make millions of copies from a single piece of DNA for a specific target. Now, in clinical applications, generally Techman chemistry is applied. So, what is Techman chemistry? Just like a normal PCR, you also over here in Techman chemistry require a double-stranded DNA and a set of specific oligos called as probes. These probes are different from the primers required in normal PCR in two ways. Number one, Um, this Tachman probes, they lack a hydroxyl group, so the Taq polymerase cannot bind to it. And secondly, on a 5' end, as you can see over here, the yellow, which is mentioned as fluorophore, and on a 5' end, you have a reporter. This is called as a reporter because it reports a signal to us as and when products are made during the cycle. And on a 3' end, which is you can see as a quencher, is a quencher and colored in black. So this quencher, it quenches the fluorescent signal and from the reporter under certain circumstances. So when we zap the probe with the light, we expect that the reporter molecule should get excited and fluoresce. But since it lies in close proximity to the quencher, what happens is the energy gets transformed from reporter to the quencher, and this phenomena is known as FRET, that is fluorescence resonance energy transfer. So, if what's important is as long as the probe remains intact, there would be no permanent increase in the fluorescence signal from the reporter and the quencher. If the reporter and quencher are separated physically during the reaction, the reporter will give a signal which the instrument can detect. And so ideally, every time the reporter and quencher needs to be physically separated, and hence the fluorescence will always increase proportionally as and when the new amplicons are being synthesized, which allows us to monitor what's happening in the reaction throughout the run. So when the initial phase, that is the denaturation step, happens, the temperature is at the highest, at the highest degree, and simultaneously as we lower the temperature, polymerization starts, the primer and the probe bind, and Cat polymerase now comes in and start building new complementary strands of DNA. But there's a probe sitting over here. So what will the Cat polymerase do? Will it go back to the position where it started, or will it stop? The answer to this question is no. Since this Taq polymerase has exonucleus activity, it will chew up the probe into bits and pieces, and hence the permanently reporter and quencher will get separated, leading to fluorescence of energies and building of new antigon. And this will be recorded by the instrument, and a graph would be plotted against it, which would be visible to us, and it is in accordance with our desired expectation. The next slide. So the workflow is quite simple. You receive the samples, you prepare the samples for nucleic acid extraction and set up the master mix and run the protocol, analyze and report. The next slide. So let's take a look at the classic example which all of us have been through, that is the SARS-CoV-2. So the SARS-CoV-2 consists of S protein, envelope protein, RNA, membrane protein, and nucleocapsid protein. So, as per the WHO guidelines, initially the E gene was supposed to be reported, followed by N gene, and a confirmatory by ORF gene and RDRT. Since these are the genes responsible for um, replication, transcription, and the host immune responses, they were of the primary importance and hence considered as the confirmatory assays. Now, viruses tend to keep on changing as they move between people during spread between people during over a period of time. And when these mutations, viruses, they differ significantly, start to differ significantly, such that the previous detected viruses could not be identified. They are termed as variants. So a continuous R&D is required for designing the primers and probes, and a rapid diagnosis is continuously required so that we do not miss any of the variants. Next. So generally the PCR cycle are programmed to run about 40 to 45 cycles. And the RT-PCR data, which is often represented, is known as the amplification plot. As you can see over here, this the colorful image. This graph consists of a vertical axis, which is in terms of fluorescence units, and the x-axis represents the number of PCR cycles. Now, this graph can be viewed in two ways. One is the log scale, as represented over here, and the second is the linear scale, which is the next thing next to this black and white graph where baseline is written. Right. So, before you interpret, analyze, and report the results, you need to keep in mind certain factors such as baseline, threshold, and CT values. So, what are these things? Have a look at them. So, what is baseline? Baseline, as you can see in the second graph, that it is defined as data from early PCR cycles before the amplification becomes detectable. Now, in an amplification plot with in a linear view, the baseline should be seen as a flat line. During this period, the baseline cycles, the target amplification might be happening, but since the signal is not accumulated enough so that it becomes detectable, it might not do so. But if the target is present, eventually it will emerge from the baseline. As you can see over here, the baseline is flat and amplification is emerging. The third thing is CT values. So
अच्छा ये जो है आप पॉइंटर नहीं दिखा पा रहे क्योंकि स्लाइड जो है ना आपके पास नहीं है। एक बार आप ऐसा करके ट्राई करिए मैडम कि अपना वीडियो बंद कर लीजिए और तब प्रेजेंट करके देखिए तो फुल स्लाइड हो रहा है कि नहीं। जस्ट एक ट्राई और करके देखिए तो फिर है ना। एक बार आप शेयर करो, मैंने शेयर बंद किया अपना।
Should I continue? Hello.
Yes, ma'am. आप continue करिए.
Okay. So can we go back to the previous? Thank you. Next thing is the threshold and the CT value. As you can see, this blue graph, which is over here in the right corner, right? So the threshold is a line drawn on vertical axis, horizontal line during the exponential phase. And during this exponential phase, the reagents are in a plentiful number. And also, this is the phase where exact doubling of the products happen. So when you drop down a line from this intersection on the x-axis, you can get a CT value and assign a CT value. So CT value is defined as a cycle number at which the fluorescence crosses the threshold signal. CT value is inversely related to the number of to the starting amount of what you have your DNA as the target concentration. So let's say, for example, I have two blood samples. A is for 200 nanograms of cDNA and B is of 25 nanograms of cDNA. So previous slide, still on the previous slide. So what I'll get the CT value earlier for the first sample, which is of 200 nanograms of cDNA. So once your PCR is run, you will get a graph something like this, which is represented over here in baseline and a no template in the blue line, a threshold, a CT value, and you should get a sigmoidal cross something like this, which is shown in red, the sample. Next slide. So the take-home lesson is, as the amount of template DNA decreases, the CT value increases. Now let's say if the CT value is too high, let's assume 35 for a 96 well plate, 96 well reaction. It will be difficult for us to distinguish between the real fluorescence and the background. So generally what diagnostic labs do is they have a cutoff value beyond which any data would be avoided or neglected. And in the software, this baseline calculations, threshold, everything is mostly done by the software in the real-time PCR instrument, and it's on the auto or default mode, or you can say on the auto analysis mode. But depending on the clinical samples, you can move the threshold lines up and down and interpret your results. So let's have a look at the real data. This is data from our lab, and this is COVID-19 data for 96 samples. Each well over here represents a individual clinical sample. So the blue lines are for N gene, and the orange is for the ORF gene, and the green is for RNSP gene. And this is in the linear scale. So for each well or each sample, you have to analyze and report in comparison to your controls. There would be a positive control and there would be a no template control. So for each single individual sample, you have to do the analysis and then report them. So next slide. So what you can see is, let's take one example, the A, B, and C. This represent a positive sample. If you get amplification for all the three genes, like in A, which is in the log scale, you can report that sample as a positive. And for B and C, it is the same sample, but I have taken pictures individually for the N gene and ORF gene along with the threshold line. The line which you see is horizontal line and the threshold line. And for the D, if you don't get amplification for the two genes, only the RNSP gene, you report that as a negative sample. If you want to have a closer look, I have full images in the next slide. Yeah, so this is in the log scale, the positive sample. Next. This is the same sample in the linear scale. You can see the amplification for N gene, ORF gene, and the RNSP. And this is the threshold set for the N gene for the same sample. This is for the ORF gene. Yeah, and this is the negative sample. You get no amplification for N gene and ORF gene. You get only amplification for the RNSP gene. So we do have certain challenges and limitations with RT-PCR because it involves complex procedures such as the nucleic acid isolation, interpreting results, setting up the reaction. So a clear-cut technical expertise and training is required to check that the instrumentation work is proper, handling of samples is proper, and the interpretation is proper. Misinterpretation of results can lead to erroneous results. Second thing is the cost considerations, especially the reagents and the instruments could be costly for a resource limited settings or for the research based where you have a limited funding. Now, the success of third thing is the success of RT-PCR really depends on the efficiency and the primer probe design. So it is advisable you thoroughly validate your assays before you procure any kits and make it into a regular practice. And the last thing is, despite its high sensitivity and specificity, RT-PCR could give false positive or negative results. False positive result may occur due to contamination during sample handling or amplification of non-specific targets, while false negative could occur due to inadequate sample collection, degradation of nucleic acid during transportation or proper storage conditions, or the presence of PCR inhibitors. Generally, the companies claim at 95% confidence interval that the sensitivity and specificity range from 95 to 100%. So, with all these things, I do want to recognize the challenges which we face every day, like how to maintain profits and generate revenues, to staffing challenges and shortages, keeping up with rapid technology advancements, and to survive in a market with competition with other labs. So, being in this service and maintaining the quality is really crucial to be in this service. Like, so at Unipath, we offer a single assay in which 32 pathogens could be detected and differentiated. Also, we have other. If anyone wishes to go, I'm showing this only for the infectious part. Else, we have a very large profile for other tests as well. If anyone wants to visit or have any queries, they can visit at the website mentioned over here, www.unipath.in. Also, we have different mini respiratory panels available, and these are in-house developed. The next slide, please. Some can detect S. aureus, some can detect parainfluenza, some can detect pneumonia, some can detect influenza. So these are various mini panels available at Unipath. And with this piece of information, I would like to stop here. So next slide. And the floor is open for question and answer. And these are my all the team members, past and present. They do exceptional work by performing RT-PCR essays, and their dedication is truly commendable during the challenging times like pandemic situation. Thank you. The floor is open for question and answer.
Yes, thank you, madam. Now, participants, attendees, if you have any question, please ask. Yes, Ankit Paul has asked one question. So I would like to ask one question regarding cDNA synthesis. What are the anchored oligo DT and random hexamers? Also, do we need to use it together or single at a time? Hello.
Yes, sir. During cDNA, what are the methodologies?
आपकी आवाज नहीं आ रही है.
So,
मेरा ही कुछ प्रॉब्लम है क्या?
random hexamer.
ओके, प्लीज कंटिन्यू. अगर मुझे नहीं आ रही है तो चलेगा, बाकी लोगों को आ रही है ना आवाज?
Yes, sir.
So anchored oligo DTs are a mixture of what do you say, like deoxythymidinic acid residues, if I remember correctly. Like you can have a DCDA or DN nucleotides. Yes, that's what I remember. And the random hexamers are like a pair of, like a stretch of primer. Hexamers like a 6 base. Hexamers means that. So random hexamers, like you have to assign random hexamers at the beginning and at the end of your primer, so that they can bind to your specific target, and it is preferable if you have a DC content, because they have a they pair by 3 hydrogen bonds, so they will bind quickly. But it should be kept in mind that DC content should not go too high.
Ankit. Yes, sir. Hello. Yes, sir. Ankit. Yes, sir. Fine. Any other participants have any other question?
Hello?
Yes.
Yeah.
Yes, Vinder.
Yeah. Hi, Varsha. It was a nice talk. I just wanted to ask how often do you validate your ashes? Is it every cycle validation you do, or is it kit, whenever you open a new kit or you get the kit from a different supplier?
Okay, so there are two things in this. Number one, when we establish any new parameter in the lab, one is that validation which we multiple profiles. Like we need to do repeatability, reproducibility, and there are many other factors like ILCP, like we have to compare our results with other labs as well, and many more profiles. Only then we can start a new parameter at our end, and also we have to get it NABL certified. The second thing is before we open, once it is into a regular practice, and before we open any new kit, for each new lot we do a lot validation and only then make it into a regular practice.
Okay, thank you.
Welcome.
हेलो,
Yes, sir.
मेरी आवाज आ रही है?
हाँ sir.
So if there is no more questions और किसी कुछ है? कंट्रोल में इसमें क्या कोरोना वायरस में कंट्रोल में कोविड डिटेक्शन के लिए क्या यूज कर रहे हैं?
सर कंट्रोल में किट के साथ एक सिंथेटिक जीन आता है जिसमें एंजीन एंड ORF होता है. सेकेंड कंट्रोल इज नो टेम्पलेट कंट्रोल एंड प्लस these are we keep in the master mix and extraction control also we do keep an internal control that is RNA-seq.
RNA?
RNA-seq is the internal control.
Okay. Ankit is again asking, your lab has this quant something app पूछ रहे हैं. Quant
Yes,
Studio
sir.
5.
Yes, Ankit, we do have Quant Studio 5. In fact, we have Quant Studio 5 DX RT-PCR. So that is a one higher version of Quant Studio 5.
So Ankit, where are you? Can you introduce Ankit ji?
एक्चुअली सर मैं रिसर्च एंड डेवलपमेंट सेल में हूँ.
आप कहाँ काम कर रहे हो?
पारुल यूनिवर्सिटी के बड़ौदा में.
कौन सा यूनिवर्सिटी?
पारुल यूनिवर्सिटी बड़ौदा.
ओके पारुल में आप. ओके बड़ौदा में ही है मैडम भी जाके विजिट करिए लैब.
हाँ सर, श्योर
गुड.
आई वुड लाइक.
And आपका eukaryotic mRNA not other because eukaryotic mRNA have characteristic of oligo A, poly A tail. So that
yes.
will work as anchor. Yes. So poly A के against में हम because if we don't know the sequence and if we wish to amplify total RNA for suppose cDNA library preparation या total RNA, then we use oligo DT primer. Because it will bind to all the mRNA है ना, tail of the mRNA, eukaryotic mRNA. Is that right, Dr. Varsha?
Yes, sir. I agree.
बैक्टीरिया में तो ये नहीं होगा. पॉली ए टेल बैक्टीरिया में तो नहीं होगा, बैक्टीरियल mRNA में.
हाँ.
नो सर.
Only in eukaryotic.
अच्छा.
Yes, thank you. So if there is no Poonam madam.
Sir, actually I don't have any right now I don't have any question for the session. It was very excellent talk, but I have some query regarding other activities related to science. Basically, ma'am, where this Unipath lab is in Baroda? In which branch you are actually?
I am in Baroda branch, and in Baroda it is located in Akota.
Akota, where it is? Near Muzmud Road?
No, Muzmud is far ahead. In Akota, nearby Radha Krishna Chowkadi, if you are aware.
Okay, I stay just near to your place, to your lab.
Okay.
I am here near to Akota Stadium.
Okay, so it's nearby the Radha.
Yeah, just in front of Kota Stadium. I am residing here. And as initially sir has just told about that, I am currently at Biotechnology Department in KPGU. Right now, under this NEP 2020, in every semester there is a requirement, or if the student want to exit for any of the year, like diploma, degree, or whatever they want to, certificate, whatever they want to go exit the course, they need a kind of certificate, this summer internship or a kind of internship. Does this Unipath lab also conduct such kind of things?
Yes, we do take students for internship programs. Yeah, many students have come over here and done the internship.
Okay, so for that, what kind of procedure would be required if we want to send our student to your lab, or how do you select the student for these kind of activities?
Okay, you send me an email or message me on LinkedIn and I forward it to HR and
फोन नंबर दे देंगे, आप अपना कनेक्ट कर लीजिएगा पुणे मैडम.
Okay, okay, sir. Sure, sir. Sure, sir.
yes, yes, that would be good.
Poonam madam was also teaching in MS University for 1 or 2 years.
For 3 sessions.
Microbiology में.
Microbiology biotechnology. I was there in from 2022, one session in 16, 17, and then 20 to 22. 2 years. For those sessions, I was there in MSU, and later on I joined KPGU. For last 1.5 years, I am at KPGU. So since our students are also requiring such kind of participation in such kind of activities for internship, 15 days kind of internship. So since this is related to infectious disease, molecular diagnostic is a part of biotechnology curriculum. So if it is something like that, that we can connect and we can look for that how we can proceed in that direction also.
Yes, sure, ma'am. Just take my number from sir and then you can message me and we can connect.
Yes, sure,
Yes.
Sure, ma'am.
If anyone has any questions, they can email me or message me on LinkedIn as well.
Yes. So Dr. Varsha is on LinkedIn also. So thank you very much, Dr. Varsha, for joining us.
I hope everyone understood at least a bit of what I wanted to explain.
Yes. And if they have not understood, so this is primary. This is like a primer.
Yes, that's why I kept all the things very basic.
पेंटिंग इज रिक्वायर्ड. दीवार पर पहले प्राइमर लगाते हैं, फिर उसका पेंटिंग होता है. सो इन फर्स्ट टाइम दोज हैव लर्न फर्स्ट टाइम, इट्स नॉट पॉसिबल कि एवरीथिंग दे विल अंडरस्टैंड एवरीथिंग, है ना. 10% लोगों को भी समझ में आया, 10-20% कंटेंट भी समझ में आया. इन दिस वे नॉलेज विल परकोलेट टू दिस सोसाइटी. दैट इज पर्पस. So our attendees are not only students but faculties also, so that even if 2 or 3 faculties researchers are attending and learning, so knowledge will percolate rapidly. That is purpose.
I would just like to know, there is one message from Vaibhavi. Vaibhavi, please message me on LinkedIn. My LinkedIn ID is Varsha Raimalani.
So I will connect Varsha madam on LinkedIn. You all should connect and share your feedback also there. Here also oral feedback you should share. You can unmute and speak. Some of you can speak and share oral feedback. Or if you are not sharing here, you should share on LinkedIn also. I will connect her, tag her, and you should share feedback. And then only you can connect with her. ठीक है? Yes, Pune madam.
Yes, sir, sure. I will connect to. Okay, so. As far as now the time for conclusion of this series lecture, so thank you Dr. Varsha Raimalani for this informative session. As sir said, no doubt this topic was very intriguing, very informative. Students are there, even if they haven't get full, but the gist of it is that they may be, actually this may be opening the door for them, that they should look for what could be possible in the infectious disease at the molecular level, how they can look for that, what they can do, because these were the basic things that were required during the COVID testing, everything, RT-PCR gene, ORF1 gene. So these all were the part of this COVID testing, and all of them are aware that this is ultimately require when you are looking for infectious disease. This was just related to COVID-19, but for others disease also. So thank you so much, Varsha ma'am, and thank you Gopalji sir for connecting us with Varsha ma'am. As for my own personal interest, also for looking for some kind of connection with her or collaboration with her regarding internship and all those. So thank you for that also. And on behalf of other the participants, so I am thanking you, Varsha ma'am and Gopalji sir. With this, I'll be concluding this session, and also, ma'am, I will be connecting you with you once Gopalji sir. I'll taking the number from him, and I'll connect you on the phone also.
Sure,
So
ma'am, anytime.
if required, I may be visiting to you also, because since we are very near to you, so we may be visiting if it is required.
Yeah, sure, anytime.
Okay.
Anyone of you can visit anytime over here if you are coming to Baroda and you're not staying in Baroda. Whenever you plan to visit Baroda, you can come over here.
मुझे तो RT-PCR और रियल टाइम को समझाने में कितना कोरोना टाइम में लग गया, मुझे टाइम है ना।
बच्चे अभी RT और रियल टाइम में अंदर नहीं ठीक से कैच कर पाते हैं।
So knowledge will percolate gradually. इसलिए इंडस्ट्री का एस्पेक्ट आपने बताया तो थोड़ा उनको लगेगा इम्पोर्टेंस। बेसिक तो जो हमारे कोर्स में किए हुए हैं रिकॉम्बिनेंट डीएनए टेक्नोलॉजी वाला, सो वी टीच 12 टाइप्स ऑफ पीसीआर। और उसमें यही बता रहा हूँ कि कॉन्सेप्ट बेसिक व्हाट इज द डिफरेंस बिटवीन आरटी-पीसीआर, रियल टाइम पीसीआर, मल्टीप्लेक्स पीसीआर, ऑल दीस थिंग्स हैं। लेकिन इंडस्ट्रियल एस्पेक्ट देखेंगे, देन दे विल रियलाइज, दे विल कनेक्ट एंड दे विल ज्वाइन, है ना, उनको पता चलेगा। सो थैंक यू वेरी मच on behalf of this Science Dialogue series platform, and we expect that people have enjoyed and learned, and gradually in this way knowledge will go. Thank you everybody, and keep in touch.
And thank you everyone. I'm sorry for the glitch that my presentation was not. I'm really sorry. Yeah.
Thank you everybody. Good night. Good night, Puna madam.